Hi. The strength of the packages, what you see in Bioconductor, for instance, Lima, HR, DESeq, is not only in what those packages can do, but also in their compatibility and availability of other packages which would work together with any of those. And in this sense, it's useful to scroll through the Bioconductor pages once in a while. You can select this keyword differential expression in search, and then you go through the list and try some out of those and see whether it has any added value to what you can do already. In this video I will try to explore a small package DEGreport, which seems to be well compatible with the DESeq2, and it has own page with all guidelines how to install and tutorial and reference manual. And if you will scroll through this user's guide, then you will see multiple references to the DESeq2 protocol. The name of the package speaks for itself. The major purpose is to do some extra quality checks and generate a good looking report of your data. The structure of the reports is pretty obvious. It consists of a long list of data, and I will not focus on it. I will only try to go through the figures and check how good and useful are they. This is the plot which looks like a histogram. It tests the hypothesis that the most of the gene expressions do not change from sample to sample, so it's a ratio of all gene expressions to the average gene. How practical it is, I don't know. This is a useful option. They hypothesize that in a good case scenario, your differentially expressed genes should not be dependent on the variance or absolute expression values. And this is the case of a good data pretty random. And this is visual of the same thing projected to a dispersion plot. Good to test with the real data. This is the plot which investigates how your factor from the sample table related to the PC scales in your PCA plot. Okay, then more importantly, whether you have some covariated factors in your sample table again. This might be important. Then more general, it can make a long QC report, well, significantly different genes you find anyway. Then another MA plot in two options with the blue stripes or without. Okay, we can test it. Looks like a ggplot style. This plot relates to the shrunken versus unshrunken fold changes in your data. It's certainly not for simple-minded users, and if you use it, please make sure you understand what it is. And my choice is to go to the next figure. Another version of volcano plot, pretty lousy in this illustration, but maybe it's much better in real life, why not? Then individual expressions of genes, yeah, useful. And the same plot, another implementation. This is a very interesting option where there seems to be a possibility to test a gender in your sample or to test whether genders reported in your sample table are as it should. It compares expression of reference genes in autosomic sort of housekeeping, then on X chromosome and Y, and obviously only male should show expression of Y chromosome. This is a little bit funny that a male has a less expression on X. It's not obvious because in case of female, we know that one X chromosome is silenced. Maybe I don't know the gender biology well enough. Then there is an option of a full report with all the figures from above. One more option is to detect patterns. For instance, what is changing from male to female in the samples. Whether it works with the quantitative parameters or not, I don't know, but we can always try. And then something more trivial, that they can recruit another heat map and combine sample data table and expression data in one. We know that it works pretty well with the P heat map. Here is another case of a complex heat map. And that's it. You can copy paste all the lines from the tutorial to your RStudio. Then you have to add some extra lines like install DEGreport. More importantly, you check what kind of data they use for the example. So they use human gender data, which at the end you can call data and see that it is relatively simple set of 21 samples, where the only column which separates those samples in groups is a gender, female and male, and that's it. So it's a one-factor experiment where design is super simple, nothing more. You can also confirm with example data that all figures can be reproduced, but it's not so much interesting for the use of this package. You need the real data. That's another story. For the real test, I decided to use this data set. It's about aging and hematopoietic cells again. The paper is published, so you can check details and compare. It has young and old human samples, and the gender is also known. The group sizes are good, and previously I used it already. For HR, the raw data are available as a separate files, which you can download and use. For the sake of time, I will try to move through the lines as fast as I can, and for details check my previous videos. First we load the libraries, then we get the data. As I said, you download them first, then check the files, then use a read.dga from HR to get them. That's, for me, works the simplest. Then this block, you simplify row names in the table to make it compatible with annotation and other business, and you get annotation. Then you get the sample information. It's a bit long story, and correct one age. The meaning is that you have to get some clean table like this with row names and all the information. Then the first figure comes. It's not only about DEGreport, it's all figures which I find useful. It's borrowed basically from the single cell analysis. You check for read counts versus gene counts in all samples and see that there are no far outliers. Very good. Here for the first time you check a figure from DEGreport whether these ratios to the average gene are okay. I don't know, so it's a bit fuzzy. Then we call already DESeq dataset because we have all the information. Note that DESeqreport accepts this sort of design, no zeros, whether you like it or not. Then you do filtering. It is necessary. Then after filtering, it makes sense to check data by box plot and be sure that distribution of all the samples is pretty random and no batch effects, no bad outliers. Then you run analysis all in once as you know it. Then you come back to this ratio to the mean gene again, whether it is raw data or normalized, and you see that it responds to normalization, but whether it is useful for the hypothesis, I don't know. Okay, then there is another mystery. If you extract your sample info after analysis, then you see this column replaceable where most of the samples are true and some are false. What are these false, I don't know, and couldn't find in the description. So I skipped it at the end for a future. Now back to the figures. We have not checked PCA yet, so now we can do it in a style of DESeq tutorial that's authentic from DESeq, or we do a glimmer MDS. Note that PCA and MDS are not identical, but they resemble one another well enough, and in both cases we see a separation between old and young groups. That's the only purpose of those plots. Now we get contrasts and move on to the MA plot. That's authentic from DESeq protocol. And now we do the same in DEGreport style, which is much better than Glimmer, because here you can specify what you're comparing with what, so you are pretty clear to yourself, and then select one out of two options which are actually the same. Never mind. And this is a DEGreport style. Note that the scale is log. It has two options, shrunken and unshrunken, which you manipulate with the raw, false, and true. So my preference is true raw data, because if you do it false, then you have those blue stripes. And next to a visual distortion, I question whether shrinking step really helps to the data analysis. Oh, sorry, my ignorance. Now volcano, it's much better than in a tutorial. It's pretty good volcano. And if you think that gene names are missing and sample numbers do not work well for you, then you can fix this as well by merging results with annotation, and then you specify in a show line what you want to show, and you want to show column symbols, and then it works with the real gene names. Actually a very good volcano. Now we finalize the data to the tops and check a few expressions from the top. These names are more difficult to fix because it refers to DDS table. It's up to you what can you do. And the same option in one plot. Okay, next. This is probably the most interesting figure in the context of this data, where you test expression on X and Y chromosomes compared to autosomic expressions of reference genes. It has to be equal. It's not really equal, but okay. Then X is also balanced sufficiently well compared to autosomic, but Y chromosome is severely disbalanced, even though you define groups by age. So it's not supposed to be like this. So here you discover that your age groups are compromised by gender, and for comparison you redraw this figure by gender. So this is clear separation of your samples by gender. Y is only in male, whereas here you have two in old group which are male, the rest are female, and three females in young, which is not enough. for the balance. So that's the greatest disappointment for this data set. Now trying to find the trends. Basically MA is extracting normalized log transformed read counts out of the data, then check design, check the data, then define your meta table as a call data format data frame, all as they suggested, but the calculation fails. And I try again a different style, fails, no luck. So now testing the randomness of your differentially expressed genes, it gives another warning that there is something not right with the low quantiles, both in terms of variance or expression. Even though we filtered data pretty extensively, there is still a bias on a low level. Pity. Then this is pure for your information what factors out of the sample table has an effect on your PC scales in PCA plot. Okay, this is more important, and it tells the same story that your group, which is young and old, is interfering with the sex. In a good case scenario it should not, but we discovered already that it does. That's pretty unfortunate, and the group is derived from age, so there is no surprise that they crosstalk. And I skip heat maps because I discussed it previously. What can we conclude here? That indeed this package is useful for the technical checkup of your data. It revealed with a crystal clarity the effect of the gender on the age groups, which is unfortunate, of course. Not all the figures are equally informative, but after all, this is a tool as every other tool. Use it wisely. Read the instruction. That's it.